So the big disclaimer, what I'm going to present is 99.9% the work of Marie-Hélène Bourget, who is on this call. So I'm only the porte-parole here. Right, so briefly gonna introduce beads for those who are not familiar with it, and then we're gonna dive into the microscopy beads specification. So let's imagine that you're a researcher doing machine learning, for example, and you receive data sets from different hospitals or institutions, and the data set usually looks something like this, where there's a bunch of files, different file names, especially like this scan final_2.ini. And at the end of the day, you want to do some like training of models, and you have to manually change the names of those files, reorganize them, and this is big wasted time. It prevents reproducibility of results when aggregating different data sets, and it's not scalable to tens or hundreds of data sets if you want to do research with a bunch of data sets from different institutions. So BITS stands for Brain Imaging Data Structure, and it solves that problem. How does it solve it? It aims at covering a lot of different imaging modalities, including MRI, computed tomography, positron emission tomography, EEG, MEG, and now microscopy. What's nice with BITS is that it has some converters to get from the raw data, which are DICOM data in the MRI world, to the BITS format. There are also a couple of metadata, and so we're gonna speak about that for the microscopy specification. And what's nice also is that the BITS community is growing very fast, which let us take on this challenge of adding the microscopy support. The fact that it's a very active community means not only that the BITS standard is updated frequently, but also people start developing tools that take BITS data as input for processing or pre-processing, but also there are some BITS data sets that are increasingly shared by the community. And what's nice is that you basically there are like a couple of repositories of like portals of BITS data sets that you can click and then download some data that are immediately in the structure that you understand. So very briefly, BITS organization is actually quite intuitive. This is sub stands for subject, so there is one subject, and there could be another session. And in that session, there could be some anatomical data. So that's an example of an MRI structure. And inside this anatomical folder, there are subject 01, session 1, and some words or like some entities that are relevant for this specific application. That's the file format that can also change depending on the modality. There is a bunch of links. I'm obviously not going to click on these links. I invite you to do it. And by the way, the link to this presentation is here, right? So it's a shortened link. You should be able to write it, or maybe Marie-Hélène can write this link on the chat. So let's dive on this microscopy BITS specification. We had to do a couple of modifications in order to accommodate the BITS standard, and these slides, will focus on those specificities. One specificity, obviously, is the file format, which is different from MRI and EEG. So there's a support for PNGs, TIFF, and OME TIFF. in terms of the modalities that are covered, these are the ones. It might not be exhaustive, but it covers probably many of the applications that are coming from the microscopy community. This is an example of an organized data set. So instead of having the ANAT, as you saw for the MRI, it's now called MICR. So that's a physical folder in which some files are organized, right? And so they could be, differentiated if there is, different samples. There could be also different stains that are used. So, that's basically how it looks like. Some comments and considerations also about the fact that in microscopy, it's quite common to acquire from one sample different chunks of data by moving the microscope at like another special location. And at the end of the day, we want to be able to reconstruct them and make sure that the chunks are associated with a particular sets of coordinates, right? So that's the way the chunks are physically distinguished across all the different files. And there are also coordinate systems for each of the chunks. I talked about metadata. We obviously spent a lot of time trying not to reinvent the wheel. We are very aware of the other initiatives, and GFS and OME and all the other, and Spark as well. So there are parallel initiatives, and we were very careful to not duplicate some efforts, but we wanted to benefit from the BEADS momentum and add microscopy coverage while also being aware of these other initiatives. So some of the metadata are taken from existing initiatives, like body part, for example, is using existing conventions for the names, and they just appear here in the JSON metadata because it's useful and it's documented in the dataset. If you'd like to have some examples of BEADS datasets, you can click on these links. So there are nice initiatives from different groups and covering different modalities like scanning, EM, like SPIM. So to conclude, there is this new coverage specifications for the microscopy data in the BEADS format. There is an article in Frontiers that has been accepted. So you can already read it. And future work will obviously focus on covering, basically exposing more BEADS data, having more tools to help converting microscopy format, like raw format, to the BEADS microscopy format. We had some discussions about how to properly create this coordinate system. We are aware of the initiatives from OME and GFF regarding the coordinate system. So we basically borrowed some of the current initiatives. We are aware that they are evolving. So hopefully the BEADS microscopy community will catch on and make sure that they are not like the developments are in sync across the different like actors of this field. So we have a lot of people to thank, people who contributed during the brainstorming sessions, during the pull requests that put together this specification. That's it. Thank you very much for your attention.
Yeah, Sandra asked me to talk about next generation file formats, which is another way of saying, how do we put our data in the cloud? Still focused largely on kind of the motivations and some of the technical details. I'm very much from outside the brain community, so looking forward to hearing what you think. I also missed the last kind of preparation talk, and I didn't watch it if the videos have been posted anywhere. So I'll go back to the first set of talks where Lee showed this slide, and I'm kind of, at least it gave me a vision of what all of you are working towards or would like to work towards. So from my side, I'm talking about the green and the yellow boxes and diving into details and kind of delineating, I guess, some of the terminology here because it does get confusing. So for everyone's benefit, I'm from the open microscopy environment, which means we focus on building mostly software infrastructure for archiving microscopy data. That means we go about, just defining some of these specifications that Julian pointed to, and try to add as many as possible, and have been doing that for about 20 years. We have this data management platform, Omero, and we have translators as well to get in and out of various formats, but I won't talk about any of that and just kind of focus on what do we do when it comes to files for today. The primary file format we offer is the OME-TIFF, again, that Julian, so a lot of this I might not even need to say, that Julian talked about. And there are some dozen to 24 microscopy companies that can export that. And it's worked fairly well over the years, but especially as we start to get into large 3D volumes, it's breaking down. And also for many years, we've been told, well, just move to HDF5, kind of by the user community or parts of the user community, and the microscopy vendors have said, no, don't do that. So we felt a little bit in the middle of everyone here. And this work on next generation file formats has, I think, given us something of a breakthrough. So where this started from our point of view is when we started getting large volumes submitted to the image data resource, the IDR. It's an international resource that runs at the European Bioinformatics Institute. The largest volume we received there was about 5 terabytes for one 3D volume. And obviously OME-TIFF just simply doesn't work. So we needed some kind of fix. And the other important driver is that EBI originally was telling us we should move to object storage, and now more and more they're telling us that we must. So actually very soon we will not be able to accept data if it doesn't live on the equivalent of S3. So we've definitely got to deal with that. Various big data formats have been developed over the years. We've looked at most of them and had gone through the various trade-offs. For the cloud reasons, these monolithic file formats like BDV and KLB simply didn't tick all the boxes. So here we're focusing on what we've come to call next generation file formats, which really means a format that's built for the cloud. You've taken the file, you've broken it up into many objects so that you can store it natively in object storage. A result of kind of breaking the file open is that often these formats are quite simple. So they're very transparent, very strong use of JSON. And that means developers have gone in and found their own way of writing down things as simple as an image pyramid, but they've done it in slightly different ways. So, a human can read that and know, okay, these are all image pyramids, but there was no specification. So that's really our starting point, was to come in and get as many groups together as we could and say, okay, let's work together and have a common way of specifying these kind of bioimaging primitives and move forward with that. What that looks like, kind of if we look into the insides of these files, on the left you have a monolithic file format like HDF5. There's some kind of hard boundary. Everything's inside of this one binary blob. There is a header. We parse the header, and the header will tell you, okay, you're looking for the highest resolution part of the image here in red. Go to this offset, read this much data, and now you've got the top left chunk of the image you're trying to show. In NGFS, you get rid of that. So there's no longer a single file. There are now many files, all stored in folders. Often you have metadata in JSON at the top. Some of these formats use something like YAML, but it's almost always very web accessible. And then you have subfolders in this case, for each of the sub-resolutions. So you have again, metadata for the highest resolution, and then you have each of the chunks of data split out into its separate file, which really means if you've read the metadata and you know what the shape and the chunk size is for the array you're working with, you can calculate the name yourself. And that really is the biggest trick of NGFS, is that you no longer need to explicitly ask, where is the data? You know how to ask for it, like ask for the URL yourself from your client application. And then you do that just hugely in parallel, and you get the throughput that you need. For what it's worth, this is kind of a side note. There is a project, so this Kerchunk at the moment, that's working to take monolithic file formats and pre-process them and make them look like ZAR. So if there are file formats that you have and you're not comfortable transforming them into NGFF permanently for archival purposes, we could talk about pre-processing, having this representation, and then maybe you can have the best of both worlds. Just something to keep in mind. Because all of this really is trying to get the speed up that we talked about in the Nature Methods paper. I won't go through this in too much depth. The downside of NGFS is that you have a very large number of files. So for the single 300 gig light sheet data set, you have 4 million files as opposed to single HDF5 files, although roughly the same size. And to note that the TIFF simply, the TIFF doesn't work. So it just completely errors out when trying to do the benchmark. When accessing these things locally, the ZAR is roughly the same performance as the H5, with the modulo that the H5 implementation is written in C++ and this is in Python. So we still need to benchmark actually using C++ against C++. There is no there's no good pure Python implementation of HDF5 as far as I know, but if someone knows otherwise, let me know. But importantly, what you're trying to do is say, okay, when we put this in the cloud, how much faster is the access of each chunk? That's the first take home. That's about an order of magnitude. And more importantly, the parallelism for an AWS bucket is based on the number of subdirectories in what you're accessing. So a single file, you can only access up to 5,000 parallel threads. Whereas if you have all your chunks spread over a tree of data, each one of those levels of the tree can have 5,000 individual threads itself. I've never written anything that can make use of that much parallelism. I guess I challenge all of you to make that happen. I won't talk through this slide. This is kind of one of the standard things when I try to explain what object storage means. I think the take home here is if you've gone this far to put your data into a cloud-based format and you have it in object storage, don't just assume, don't stick with your common access patterns of just downloading all the data. The whole point is that it's been broken up. Your clients can now access individual pyramid levels, and you can save yourself a lot of bandwidth if you teach your clients to only access what the user's interested in. So just keep that in mind, is that download is really no longer your best friend. The caveat to that is that means our clients then need to be written differently to make use of all of this throughput, and clients must must use the parallelism, and they must be aware of their chunk boundaries. So that's the downside, is that we really need to work in a different way. Okay, I know I'm running out of time. Sorry, I still got a bit more to do. What we focused on really is building clients across a number of languages, a number of ecosystems. This is VZAR in the browser, but there's also Nepari and Fiji plugins. If there's a different ecosystem, I know Matt's working on ITK. If there's anything that's not represented here, that would be something that would be good to hear from all of you. What would it take to make NGFFs useful to you? The other is that the process for the specifications is all happening on these links, like with bids. And we're just kind of moving through. So where there are specifications that you think you need to make NGFFs useful to you, it would be great to hear about that. And the goal is really to prevent multiple file formats if we can. Obviously, there's no way to prevent file formats from being written, but if we can agree on something, that would be wonderful. And this is kind of the last bit, going back to Lee's slide. And this is a little bit more hypothetical, is that I wondered to what degree NGFF shouldn't be something that OME is doing for microscopy, but if we can't think of it as just a imaging standard in the same way that TIFF is. So if we look at just an OME TIFF really is just a very, very thin layer. What it takes to turn a TIFF into an OME TIFF is to add the OME XML metadata and then name the file .OME TIFF. That's all it really means. The there's a bit of a downside in TIFFs that you can only add metadata in one location, currently in the image description, and different applications like Micromanager, we disagree on how we're going to store that. If two ecosystems want to own the file, what do you do? I think with the NGFF, since it's hierarchical, we actually have the potential of going, okay, well, can we have a multi-ecosystem file format? So you take the ZAR, you put the OME XML somewhere. From my point of view, that is now an OME ZAR and OME NGFF. That's great. Could we store, other metadata, maybe a graph of JSON LD that is cross ecosystem? That would be great. Or maybe just as a separate location you have, I don't know, here I'm completely out of my wheelhouse, bids metadata or some other metadata that you need, all in the one file set. And so that we're working together on the NGFF to create just an imaging standard, and then each community can adapt it as it needs. So I think that's potentially useful to the Brain community. I actually hope that goes further and could be of use to the DICOM community. They don't currently have a cloud-based format, so if anyone has connections there, it's something I would love to talk about. There are other projects that are ongoing, like bioschemas, that we could use a common vocabulary, again, so that each of our projects isn't needing to define specifications for how to talk about proteins, let someone else do that work. And then there's even the geo community that's using, not the metadata, but all the other concepts I've talked about are happening in the geo community. So again, I think there's a lot of room for overlap and kind of expanding this into common usage patterns. I'll shut up. Thanks.